Welcome everyone. We're going to go ahead and get started with our talk today. So I'm very happy to introduce a new faculty member here at UT Dallas who's joined us just this fall. So Dr. Jerillyn Kent received her bachelor's degree in psychology and biology from the College of William and Mary in Virginia, and then did her PhD work at Indiana University in clinical psychology. She then went on to complete postdoctoral fellowship at the University of Minnesota, and there she was focused on understanding motor abnormalities in the context of psychosis. So as I said, Jerry joined the Department of Psychology just in 2021. We're happy to have her here. Kind of generally speaking, she investigates neurosubstrates of motor abnormalities in the context of psychopathology, and then is exploring the link between those abnormalities and various clinical phenomenology that are correlated with it. So today she's going to share with us some work on cognitive control, from motor inhibition to novel therapeutic intervention. Welcome, Jerry.
Thank you, Karen, for that terrific introduction. I just want to say how happy I am to be here at CVL for a science luncheon lecture. And with that, I will go ahead and dive in. Okay, so a roadmap of where we're going to go today. First, I want to just orient you all a little bit to my broader program of research. As Karen said, investigating motor abnormalities in individuals with psychotic disorders, and how the work I'm going to talk about today fits in with that broader program of research. Then I'm going to do a deep dive into an EEG study of cognitive control and motor inhibition in individuals with psychotic disorders. And finally, I'm going to talk about the implications for treatment that are sort of adjacent to some of the work that I'll be discussing today and some of those constructs. Okay, so as a brief introduction to make sure we're all on the same page about the core features of psychotic disorders, this is the clinical population that I study. Most people think about symptoms like hallucinations and delusions, and those are very important core features. We also see what we would call negative symptoms. So this refers to symptoms like decreased emotion expression, decreased motivation, decreased social engagement. Finally, there are impairments across a wide range of cognitive domains. So domains like working memory, executive function, cognitive control. The other aspect of my research is that I sometimes investigate these phenomena across what we refer to as the psychosis spectrum. So I'm going to refer to it in a particular way today, but just so you know, this can mean a couple of different things depending on context. So the first thing the psychosis spectrum can mean is it can refer to a group of diagnostic categories that you may be familiar with, like schizophrenia or schizoaffective disorder, that share those core features but have some minor differences. So sometimes we refer to the psychosis spectrum as a way to think about all of those psychotic disorders that share core features together. Relevant for my talk today, we can also think about a psychosis spectrum in terms of a genetic liability. So biological first-degree relatives of individuals with a psychotic disorder share 50% of their genetic material with their affected relative. So we can think about them as being on a spectrum of genetic liability for a psychotic disorder. Just for your information, I'm not going to go into these types of results today. We can also think about the psychosis spectrum in terms of phenotypic expression. So there are various groups of folks who present with subthreshold psychotic-like symptoms, so symptoms that are similar in content but lower in severity to the core features of psychotic disorders. So we can think about there also being a spectrum in terms of intermediate phenotypic expression. Okay, so to orient you to my broader program of research looking at motor dysfunctions in the psychosis spectrum, the great majority of this work is focused very specifically on the cerebellum. So if there are any repeat customers from my talk on Friday, I spent a lot of time talking about my work here in this area. I've used behavioral and neuroimaging measures to investigate cerebellar integrity in the psychosis spectrum. This was mostly my graduate work. And then as a postdoc, I moved on to interrogate other aspects of the motor system. So you can see here as an example, I looked at movement fluidity using a tablet to look at handwriting fluency. That's thought to be more related to the basal ganglia. I used EEG to look at motor inhibition. So, exploring the construct space more thoroughly. Then my current and future directions really are focused on trying to translate this body of work into potentially interventions or intervention relevant work that may actually work to improve symptoms or functioning in this population. So for today, something that I thought would be of particular interest to the group of folks here at CVL is my study of motor inhibition, this EEG study, potentially given the very close relationship of motor inhibition to cognitive control, which, as you all know, is super important for aging. So before I dive into this study, though, I want to take just a minute. Oh man, glitchy mouse. Okay, good. It's going to be a task. Thank you for laughing. Okay, so I want to take a quick minute to acknowledge Abe Van Boers, who's the first author on this manuscript. I supervised Abe in the conducting of this work when he was a postbac, very talented postbac, and now he's a medical student. So I want to make sure to acknowledge all of his hard work on this paper. Okay, so let's talk about the broad construct of cognitive control before we get into the specific instantiation in terms of motor inhibition. So I probably don't have to tell you all, but very broadly, at the broadest level, cognitive control refers to the ability to behave in a goal-directed fashion. Being particularly important in instances where there's a prepotent inhibition, or maybe a prepotent response, or there may be competing responses, or maybe there's something you don't want to do, but you want to because of your goals. So we think about cognitive control as being particularly important in those sorts of contexts. Cognitive control can be thought of as being composed of both proactive and reactive components. So the classic proactive cognitive control example is when you want to go to the grocery store after work. And so instead of just taking your normal path home, you need to remember to take a different path and go to the store. So in proactive cognitive control, this would be the situation where you're thinking about it all day, thinking, okay, I gotta go to the grocery store. Thinking about it at work, you go out to your car and you're like, don't forget that grocery store, maybe even while you're driving. And so that lets you, representing that goal proactively, will then hopefully result in you taking the alternate route, going to the store, mission accomplished. Reactive control would be something that shows up when, for whatever reason, you were not maintaining the goal throughout the day. So you went to work, you taught, you went out to your car, you're driving home, and it's not in your mind, but then you see something that cues you to remember this goal. So maybe the turnoff to go to the grocery store, and you're like, oh, right. And so the reactive control is responding to that sort of cue, reminding you of your goal, and then being able to act on it instead of what you were going to do. So that's sort of the illustration of the broad construct that we're working with here. So when we think about motor inhibition, so what this is a response you've already started making, and then you need to inhibit it. So that is something that absolutely requires cognitive control. Motor inhibition deficits have been widely reported in schizophrenia. So this is a particular psychotic disorder diagnosis. And then there's not as much work that's been done on biological first-degree relatives. So in this case, the investigation is inquiring as to whether motor inhibition deficits are associated with that genetic liability that we talked about. So there isn't a whole lot out there. And the behavioral findings are sort of mixed as to whether there is this behavioral deficit in motor inhibition. And there is one study showing evidence of preserved behavioral performance, but abnormalities in motor inhibition relevant neural activity. So what we wanted to do for this study was to dig more into this open question of whether there are motor inhibition deficits in biological first-degree relatives. And we were thinking about this in the context of even in patients where there's been more work. Most of that work and that one study in relatives that I referenced uses fMRI. And in this case, certainly you can get some temporal information about fMRI, but when you're thinking about a very complex process like cognitive control that has multiple different stimulus and maintenance and response type components, you're really mostly getting information relevant to where the deficit is when you're using fMRI. And so we thought in addition to further interrogating this open question in relatives, we can use EEG, which has a much finer much finer temporal resolution to really investigate like which parts of the cognitive control process are behaving abnormally, and that can kind of tell us more about how cognitive control in general is dysfunctional in this population. Okay, so how did we look at motor inhibition? We used a stop signal task, and what that is for the most part a very simple reaction time task. So we have a series of white fixation crosses. After a small amount of time, the one on the right or the one on the left turns into an X. And all your task is to respond with the corresponding thumb. So if you see the X on the left, it's a left thumb press, right, it's a right thumb press. And you can kind of see how we like build up and put some pressure behind this prepotent response. This is a very natural thing to do and people can really clip along. So the stop signal task is mostly this, except sometimes you might see a stop signal. So what that stop signal is in our study here and in other studies, a set of white boxes around the original stimuli. So on these trials, you would see your fixation crosses, you'd see the cue to make a response, you'd start making the response, and then you'd see the stop signal. You have to not make that response after all, even though you'd already started making it. How do we know that they've already started making it? Well, basically, the latency for this stop signal to show up is titrated between participants, but also within, across the session within participants, so that behavioral performance hits around 50%. So what that means is the stop signal is showing up so quickly that you really can't inhibit the response about half of the time. So this is a very frustrating task, and I'll tell you just a little bit more about the structure of it next. So when we're running a stop signal task, we start with 20 trials where there's never a chance of needing to inhibit a response. It's just that simple reaction time task. There are then 120 trials that are basically the same, except 20% of the time you see the stop signal, and you're supposed to try to inhibit that response that you've already started making. We end with another 20 trials where there's never a stop signal, and this is an EEG study, so we need a lot of trials. So we do this for 5 blocks. It's a lot. Okay. So you may be thinking, okay, so then what is your behavioral measure? Because we've sort of set it so that accuracy is always going to be 50% for everybody. So what we're interested in is the amount of time it takes to successfully deploy the inhibitory processing necessary to inhibit that response you've already started. I don't have to tell you that it's really non-trivial to try to measure the latency of a process that's characterized by a literal lack of response. So that's kind of a whole big thing. So what we've done and other folks have done is to do. Well, I think we're a little frozen. Uh-oh. Maybe I will just escape. Oh, hello. Nice to see you. All right. So yeah. So the next thing I'm going to do is tell you how we estimate or hack this idea of how long it's actually taking to deploy the inhibitory response. Okay, so what do? So the first thing that we do is we take the reaction time distribution for participants. So we're looking at all of the reaction times that participants are providing when they're actually making the response, but there could have been a stop signal. So we look at the distribution of all those reaction times, and we take the actual reaction time that corresponds to the percentile of behavioral performance. So remember, that's about 50% for everybody. So we take the 50th percentile reaction time within this distribution, and we take that number, and then we also look at the number that corresponded to the variable delay. So again, this is the delay between the signal to make a response and the stop signal. That's titrated for everyone to make sure we get that 50% accuracy. We look at that particular delay, we call that the stop signal delay. And using those two numbers, we're able to calculate an estimate for how long it takes to successfully deploy the inhibitory processing when people are able to inhibit a response that they've already started. So just to break this down a little bit more in terms of like how it's grounded in how we're thinking about the task, if you think about this 50th percentile reaction time, you can think about the fact that half of the time, the reaction time will be faster than that 50th percentile reaction time. And in that case, that reaction time wins out over the inhibitory processing reaction time. We call it the stop signal reaction time. And so half of the time, the reaction time is faster, it wins. Well, that kind of maps onto that behavioral 50% accuracy. Again, using that 50th percentile reaction time, half of the reaction times will be slower, in which case the inhibitory processing reaction time will win, and you're actually able to inhibit that response. The inhibitory processing kicks in, and you're able to inhibit the response because your reaction time is going to be a little bit slower that time. Okay, so just to summarize, this value here, stop signal reaction time, is an estimate of how long it takes to deploy inhibitory processing in this context of motor inhibition. Okay, so this is what we're looking at for our behavioral measure. So again, I mentioned we were interested in looking at the neural correlates of motor inhibition in individuals with schizophrenia. We also had a sample of their relatives, their biological bursary relatives, and a control group. What I've plotted here on the y-axis is that inhibition reaction time that we just talked about. It's in quotes because you're not actually responding. Okay, so what we see here is a very robust effect in patients. So a longer time to deploy the inhibitory process that is less efficient, less optimal situation. So we would say this is in line with other evidence of inhibition, motor inhibition abnormalities in individuals with schizophrenia. We see a very striking lack of effect in our relatives group. So this is sort of a point in the column of preserved behavioral performance when it comes to motor inhibition in biological first-degree relatives. Okay, so the next thing I want to get into are our EEG findings. So just real briefly, when you're taking an EEG, you're measuring electrical activity at the scalp. It's coming from the brain, but it is, the signal is very small. There's a lot of noise. Of course, we do our best to take out any systematic noise sources like muscle artifacts and such. But on the level of an individual trial, the signal is very small and noisy. So when we want to look at EEG activity that corresponds to different aspects of a task, for example, stimulus presentation, the response you're making, we have to average that EEG activity over the course of many, many trials. And when we do that, we start to see a systematic neural activity that is linked to stimulus presentation and response, and we call that an event-related component. So just for the sake of illustration, here's one now. So you can see here, when we average, we start to see this coherent neural activity that emerges when we average, in this case, based on when the stimulus is presented. So we looked at the P300 specifically, so I'm going to talk a little bit about that now. So what we see in the P300 is when we anchor the averaging to stimulus presentation. This depends on the task and other parameters, but by and large, you'll see somewhere in the neighborhood of 300 milliseconds after stimulus presentation. You can see our window for this particular component here shaded. We see a positive deflection, peaking around that 300 millisecond neighborhood. So like I said, it really depends on the particular task and context, but by and large, P300 is thought to relate to processes like attention and task relevance. People also think about it in terms of salience, the salience of the stimuli in the context of the task. Okay, so what did we look at with the P300 and what did we find? So essentially, we were looking at how the P300 component varied as a function of whether or not you might have to inhibit a response when you saw that cue to make a response. So to break it down here, essentially we're looking at the P300 in response to the stimulus to make a left or right hand response. And we compared it in situations where there was never a need to inhibit a response versus situations where they still made the response, but you might have actually needed to inhibit it. The actual stimulus is exactly the same, but it sort of means something different in these two conditions. All right, so what did we find? So what I want to do is just first talk about a general pattern that we saw. So you can see this frontal central electrode. We saw a modest P300 peak in response to the stimulus that said, okay, make a response, and there's no possibility of needing to inhibit. And by and large, that peak was suppressed, and the amplitude was lower when that same stimulus showed up to elicit the same response, but there was the possibility of needing to inhibit a response. And so the way we're interpreting this is that this is really sort of in line with this idea of P300 as carrying some salience about what it means, but what the stimulus means, because in this case, you're getting an X, you need to respond. It's very straightforward. You're just responding. It means something very clear, and there's a very directed action to take. Here, the stimulus is the same, and so is your response, so what it means is different. It means you should respond, but that maybe that's not going to end up being the real response. And so we're thinking that this P300 modulation is a function of the possibility of needing to inhibit, is related to the fact that it's just not as clear what this task actually means in this context, or what this stimulus, actually means in this context. Interestingly, you can see our schizophrenia patients in red, and I should say we did have a small bipolar disorder sample, but I'm not going to focus on those results today. That sample wasn't as well powered as our other groups. We did not see this modulation in our schizophrenia patient group. So here in red, P300 peak, and unlike the other groups, does not suppress as a function of the possibility of needing to inhibit a response. And the way we're interpreting this is through this idea of this being like a proactive inhibitory mechanism. So you're seeing a stimulus to respond, but you're thinking, I don't know if it's actually going to go that way. So I'm kind of like keeping this goal or this idea in mind that it might not be like really how it's going to go. So the lack of this modulation as a function of potentially needing to inhibit a response is suggesting to us that this is some sort of proactive inhibition deficit related to not sort of updating the context of the fact that like, the stimulus means something different now that you might actually have to inhibit a response. Okay. All right, so the other event-related potential I want to talk about today is the lateralized readiness potential. So very broadly speaking, what we're interested in here is the idea that when you make a movement, say with your right hand, there is, obviously that is instantiated through neural activity of the contralateral motor hemisphere. The idea with the lateralized readiness potential is that any premotor activity should show up specifically on that contralateral side. And that's what we can see. Again, this is just like a broad level idea here. When we look at neural activity preceding a response that is specific to the contralateral hemisphere, so this is a subtraction wave, we see a negative going event-related potential. Again, this is prior to the response, so this is thought to reflect motor preparatory neural activity. Okay, so there are a couple of different ways you can calculate the LRP, and those different ways afford you different lines of inquiry. They tell you different things about what's going on in terms of premotor neural activity. So in this example here, we have computed the LRP by averaging the EEG signal anchored to the stimulus that cues the movement. Maybe I shouldn't say cue, but anyway. So you see the X, you're getting ready to make a response, and this is all these are all trials where a response was actually made, I should say. And so you see the stimulus, you're going to make a response. When we average the LRP anchored here, we start to see that negative going ERP that reflects premotor activity. So what we can look at when we are looking at the stimulus locked LRP is we can look at the time between stimulus onset and the onset. So we calculated this at 50% amplitude. So the onset of the LRP, and so the onset of motor preparatory activity. The idea here is that this intervening time, this onset latency, reflects processes like response selection. So you have to decide like which thumb you're going to use, and the activation of the motor response, so that initiation of premotor behavior. And that's what we can look at, how long those processes take using stimulus-locked LRP. So I'm only showing you the stimulus-locked LRP from one condition because we did not see any kind of condition effect with this. So the time it took to select the response, to initiate the motor preparatory sequence did not vary as a function of whether or not there was going to be a need potentially for inhibition. We did see a significant group effect where these processes were slower in our schizophrenia group, and this is in line with other literature that's shown this before. So this is not relevant to our task particularly. Yes?
Oh, please continue with the sentence.
Oh, sorry. Very salient. So yes, this is not relevant to our task manipulation, but it is sort of an interesting way to understand, some of the motor type processing. Yes.
This is very interesting to see that. So from what I understand, it seems that the schizophrenic, patients are not having a later initiation, unlike the bipolar disorder, because they seem to have a shift. But they just have a more attenuated N200 response, right? Unlike the bipolar disorder, which you have a shift. I mean, it's not the amplitude. It's like more like a shift.
I'm so glad that you asked. So these plots are so they're so coarse. Because if you think about everything that we're averaging, and it's also a subtraction wave, so we're just looking at the difference between left and right hemisphere. So
They were not significant.
It doesn't look like, so this actually wasn't significant, this amplitude effect, I know. And this actually was significant, this red versus blue and purple. It doesn't look like it because we've grand averaged across everybody. And so I can't tell you exactly where that onset latency is. I mean, obviously we look at the butterfly plots to make sure it's what we think is happening. So we're plotting all the components for everybody. But yeah, it's somehow this was significant. And your note about the bipolar disorder is correct. They actually were significantly later than the relative group, weirdly, I guess because of different variances. But again, that sample is kind of small, so I don't want to make too much of it at this point. Yeah, so even though it doesn't look like it, there was a delayed onset. And actually, when I go into the response-locked LRP next, I'm not even going to show the waveform. I've got it if anyone wants to take a look-see afterwards. But yeah, you almost just have to look at the actual numbers in terms of onset latency, which I didn't do here because it didn't really vary as a function of the process we were interested in our task. But thank you for giving me the opportunity to clarify all these complexities. Okay. All right, so the next component that I want to talk about is also the lateralized readiness potential, but in this case, it's calculated by anchoring that averaging to the actual response that was made. So when we look at the LRP this way, What happens is, so here's the response that was made. The LRP activity is this negative deflection, again, in the ERP that happens before the actual response. So again, we're thinking about motor preparatory activity, and what the response locked LRP can tell us, if we look at, again, that onset latency, which is a little wacky to try to actually look at here, but if we look at onset latency with respect to the actual response, this latency value, that can give us a sense for the time involved in response execution. So how long is it actually taking from the time that you initiate the preparatory activity to actually making the response? That's what we're looking at here. Okay, so again, what is happening in terms of our contrast of interest, this is really similar to the P300. We're looking at that response locked LRP and how it varies when participants are responding and there's never a need to inhibit, versus when they were responding and they still responded, like we needed an LRP, so they responded on all these trials, but they might have had to inhibit the response. Okay, so what I'm plotting here is that onset latency that we were talking about. So 0 corresponds to the actual response time. And so these negative values then reflect how many milliseconds before the response did that neural activity corresponding to motor preparation onset. So just to ground this a little bit more, you can see here in the red bar, this reflects an LRP that onsetted about 140 milliseconds before the response. And the blue bar onsetted approximately 120 milliseconds before the response. And the important thing here is that the responses summarized by the red bar are longer. They're taking longer to execute these responses. So the time from the initiation of the motor preparatory activity and the actual response is longer. The response execution is longer in the red bar versus the blue bar. And what this means in terms of our data is that in our control group, when it was there was the possibility of needing to inhibit the response, that response execution time was elongated compared to trials where they were just responding and there was never a need to inhibit a response. We did not see this similar modulation in the relative or the patient group. So this is really interesting to us because not only is it another piece of evidence for this preserved behavioral response, but some kind of neural abnormality associated with inhibitory processing in relatives, it's giving us a really kind of clear picture of what exactly that abnormality is reflecting. So this failure of response execution modulation as a function of potentially needing to inhibit the response. And so again, this is emerging as a proactive inhibition mechanism. So you're making a response, and you're not consciously doing this, but you're starting it, and there's kind of something about it where it might not actually go through. So that's what we think this is reflecting here. Okay, so I want to just briefly summarize what I presented today. It's a very complicated study. So we saw a robust evidence for motor inhibition and dysfunction in our schizophrenia patient group. And that was evidenced by taking a longer time to successfully deploy the inhibitory processing. And that was quantified through that stop signal reaction time. So again, estimating the time for inhibition through looking at the stop signal delay and the distribution of reaction time. We did not find evidence of dysfunction on a behavioral level in our relative sample. So again, that is a point in the column of a preserved behavioral response in biological first-degree relatives of individuals with the psychotic disorder. Okay, so we also found only in patients evidence of neural abnormalities in inhibition-relevant stimulus processing. So That was that failure to modulate P300 amplitude of a function of potentially needing to inhibit a response in response to that signal to make a response. And again, we think this reflects this idea of salience that, in our other groups, there is different meaning behind the cue to make a response when you're not going to need to inhibit it versus when you are. And that failure to modulate that in the patient group is demonstrating some sort of proactive inhibition dysfunction. Okay. Next, we found failure to modulate response execution time as a function of maybe needing to inhibit the response in both patients and relatives. So again, what we saw based on the response-locked LRP was that our control group was elongating response time. So the time between the beginning of motor preparatory activity and actually execution of the response, when there was the potential to maybe need to inhibit the response. We did not see that modulation of response execution time in our patients or our relative sample. So there is, again, evidence in the column of a preserved behavioral but abnormal neural inhibitory activity in relatives. Its emergence in relatives is suggesting this is associated with that genetic liability for psychotic disorders that we talked about earlier. And what's interesting here is that the dissociation in terms of where the result shows up as, or where there's a positive finding in relatives, it really gives us information about the nature of the inhibitory abnormality. That was why we wanted to use EEG to be able to decompose different aspects of the tasks, processing, response preparation, etc. Okay, so the last thing that I want to spend just a few minutes on are implications for treatment. So essentially, how can work that is adjacent to the work that I've just talked about potentially be harnessed to actually intervene and improve functioning related to cognitive control? Okay, so I mentioned at the start here that motor inhibition deficits are widely noted in individuals with psychotic disorders. The same goes for cognitive control impairment more broadly. So there's lots of evidence for cognitive control dysfunction in individuals with schizophrenia. There was a recent study, it was a pilot study, and some of the behavioral findings are a little messy, but by and large, the punchline is that it seems like neuromodulation was able to increase cognitive control in individuals with a psychotic disorder. So the thing I'm interested in here is that this construct that I've been studying does seem to be a potential intervention target. So not just specifying the nature of the dysfunction, we might actually be able to target it to see some improvements in that domain. And then obviously, given how important cognitive control is for lots of things, but just very generally, behaving flexibly in a changing environment, adaptive behavior, we think that could have some real downstream consequences. The last thing I want to say, and this is a literature I'm just dipping into, and it seems, as everything in psychology, long story short, it's complicated, but it does seem that even though the source of the cognitive control dysfunction may be different, there is quite a bit of data showing decreased performance on cognitive control tasks in other patient groups. For example, depression and anxiety. And so this is really exciting to me because this transdiagnostic emergence of cognitive control being implicated across traditional diagnostic categories is really in line with NIMH's Research Domain Criteria initiative, where they're really wanting to look at processes that cut across traditional diagnostic categories. The fact that we may be able to intervene on one of these transdiagnostic processes and have that actually help folks with many different types of clinical presentations is also really exciting, and I think could be very impactful. So With that, I want to go ahead and acknowledge the folks that I worked with on this paper. So this is a part of my postdoc, so I want to acknowledge Scott Sponheim, my postdoc mentor, senior author on the study, and then Abe Van Boers, first author who I supervised in doing the study, writing the paper. Our other co-authors, obviously our funding sources, and thank you all very much for your attention, and I'll be happy to take questions. Thanks so much.
Question.
Yeah.
Do you have any more general neuropsych measurements on your participants for the study, or no?
That's a really good question. So the answer is yes. Scott's, just in general, Scott's samples are very thoroughly phenotypically characterized. For this study, so I want to be really careful about, we actually did report some correlations in the study, but they were very exploratory and didn't correct them, so I didn't really want to get into them here because it kind of like makes me nervous statistically. So I don't remember what we looked at specifically in terms of neurocognitive or neuropsych. I know that we didn't find significant associations. So it's possible that there's more that we could look at. What did we find in terms of correlates? And we found some stuff, but like I said, we didn't correct for multiple comparisons, and there were a lot of comparisons, right? Like we had P300 modulation, P300 amplitudes. It may correlate with subthreshold psychotic symptomology, but I don't want to say it because I would be a little worried, just given all the correlations that we did. But people want to know, so we looked, and there's maybe something there. I didn't see any neurocognitive correlations, but we may have just done a gross measure like the WAIS or something. So it could be that we could have looked with domains if we had a real specific hypothesis.
So I'm wondering about processing speed specifically. If you had a measure of processing speed to check to see if they're equivalent across the group, or if you did have it, if you take that into account, would that then influence the timing?
Oh, that's such a good question. Okay, so we definitely have that, and there would definitely be differences. If I'm just, I would expect there to be differences in our patient group. I don't know about relatives. So let's think about that. So are you thinking that could be relevant for, certainly for that stimulus locked LRP, where we're seeing slower response to execution and activation of the preparatory motor pressure behavior? Do you think it would, are you thinking of other components?
This is outside,
Really
this literature is outside my expertise, but I was just wondering, it would be nice to show that the schizophrenic group didn't really differ in processing, even something that simple, compared to others, because then if they did, does that change your inference? Maybe it doesn't, I don't know, it's just something I was thinking about.
That's a really good question. So let me try to speculate here. I know they would be different. So I do think that it's absolutely playing into this result that I was talking about, agnostic to condition, with respect to slower response selection / activation of the motor response. So I think it's definitely playing into that. In terms of the P300 results, we didn't really find anything with latency. So, that indicates to me that is more tied to this different kind of salience of those cues, varying as a function of the context they're in, whether you're going to definitely make a response for sure, or whether you're going to make a response when we might throw up one of the stop signals. It's so annoying. So yeah, so we're there. So let's think about response execution time though. In fact, maybe I'll even go back to that graph. So that was what I was most interested in, coming from a motor background in these data sets. Yeah, response execution. I honestly think, and I would have to really wrap my head around it. So I guess we're not really seeing it there. I'm speculating here. So I need the latitude. So latitude please, but I would honestly be potentially more concerned about, and this is related to processing speed potentially, but psychomotor slowing on a behavioral level, which we do see in individuals with psychotic disorders, kind of contaminating that. And I don't see that here, but that's a really good consideration that we should take into account.
Thank you.
That's a great question. Yeah, well, sorry.
Oh, no.
This is just a follow-up Karen's question. One of the simplest trials to look at would be a simple choice reaction time for processing speed. which would still have the two-handed response, and then you have, let's say, a green and a blue color, and you just have to press one over other. If you have something like that, it's hard to understand from here if the go reaction time is capturing the processing speed, because it's not, because the horse race model, the way these, stop RT tasks are designed. So you get faster at go, and then your stop, trials become faster. So sometimes as participants, we try to slow down the go RT. And so there is a lot of planning also that you can use in these tasks, and we don't know whether some of these patients are trying to slow down the go to make sure that they are able to do the stop very well. And yeah, anyway, so just based on this task, it's, again, for me, it was like, is it really proactive control, or like, is it failure of proactive control, or is it like executing of a strategy? Because every time I do stop RT, I try to slow down my ORT so that, I don't get, stitched on the
oh my goodness.
Stop RT.
That's such a good point. Okay, so I have a couple of things that your comment is making me think of. So, we try to account for that. It's, sometimes you see a stop signal and it's like rigid, which, is obviously like highly vulnerable to what you're talking about. So what we try to do, and maybe actually, because this isn't like give y'all a headache, maybe we can go back to this thing here and think through this together. We try to account for that by anchoring this calculation in the reaction times that are showing up when there might be, like what you're talking about, when there might be an inhibitory response. And like, between using that as our reaction time anchor and this stop signal delay that's supposed to get to 50% accuracy, like theoretically, we should be able to account for some of that stuff. But what ends up happening, which we definitely saw, is that people just don't make the response. Like, it's exactly what you're describing, where they're like, oh, I'm just going to hold off. So we tried to mathematically correct for that. So we called those errors of omission. And I haven't thought about this math in a really long time, but we did do something to try and shift where on that distribution we actually picked from as a function of how much we were seeing that behavior. So like in actuality, maybe we actually chose like, anything about this ability response. Yeah, so if we thought that this 50% accuracy wasn't really right, like it was just because people just weren't doing it, then maybe we would pick like a value over here. Like that's not perfect. And that was like a whole big thing that we had to control for. So yes, those are very good points. And we absolutely see them playing out in our data.
Yeah, I mean, I was not, changing the task is a great concept, the classic task. What I was saying is that the Go RT is not giving us information about processing speed. And so we really can't answer the question about processing speed, which is a great question. So I'm suggesting maybe a task like having a classic twice reaction time task, just to add it to it, to see if that contributes to the stimulus response relationship, to what extent that kind of simple motor control contributes to this kind of.
That's a great idea, particularly because when you said processing speed, I immediately went to like a neuropsych, like digit symbol, and that's so much more grounded in what we're actually concerned about. So yeah, that's a great idea. Thank you. Kristen.
Yeah, I hate to move you all around. That's why I tried to do my question.
Oh, no worries.
The one where you showed the difference in the bars, the red. Yeah, the red and the blue.
These are dense. There are actually not that many slides. There's just a lot on each slide. Okay.
Yeah, this is just me thinking. I was thinking that your design across the trials actually did control for a lot of these things. Like I think it controlled for kind of differences in just baseline kind of speed type things as you're making this metric. So that made me think that this was kind of directly interpretable then. So this is sounding way more complicated as I said it than it did in my head. And so it seems like that would be a nice within subject in individual difference variable. And so if greater distance between blue and red is good, then you have this how small that difference is in the psychotics, how small that difference is in the relatives of the psychotics. Could be some interesting individual difference measure that if you computed it and it had enough variance in it, maybe you could use that to correlate with other things like outside of this study, like as you move forward, like do those people have poor dopamine binding in PET studies or genetically, or you know what I mean? So if you want, if you want to kind of then dig into kind of mechanistically what's driving this, if you had this individual difference measure, which I think this would make a nice one, then outside of this study, you could kind of compute that and use it, not mechanistic with a small m.
Yeah.
Right? I don't know.
So that's a great point, and it's so funny that you bring that up because we actually did compute the difference in the P300. Like we did a modulation index, and that seemed to potentially track with that subthreshold psychotic symptomology, but I don't want to oversell that. I don't think we actually did that here, which is a little weird because it's kind of like the whole thing. And I think we looked at it and we were like, oh, there's nothing there. But you're right, there's got to be variability just like in that P300, which we could look at in relation to all this like rich phenotypic characterization. I'll let you know if I'm wrong and we actually did, but I think I would remember it. That's a great idea.
If it measures something really specific, then maybe this is an index of kind of premotor function versus like a frontal striatal, or like an anterior cingulate, because I'm thinking about like monitoring the situation, inhibiting, that's a very like limbic kind of like reward, and so I'm just wondering if this is kind of a more pure like premotor type thing. I don't know.
Right, like it's like those premotor area talking to like motor execution, like kind of exerting the regulation. That's really interesting. Yeah, thank you. I would get lots of good ideas coming here.
Do we have any online questions, Camilla?
No questions at this time.
Okay, thank you. What about from the room? Any last questions or thoughts? Hey, thank you so much for an interesting talk, Jerry.
Yeah, thanks so much for having me. I really appreciate it.